My name is Sun Yuan. I'll introduce the paper submitted to iMeta. The title is Spaceflight Redefines Aging-Associated Microbiota. Space exploration is a major human achievement. As space travel becomes more common, studying its effects on the human body, including microgravity, radiation exposure, and isolation, is critical to addressing both immediate health risks for astronauts and long-term consequences for humanity. Spaceflight disrupts the immune system, causing inflammation and dysregulation, and alters gut, oral, and skin microbiota, which regulate immunity and aging. This study examines how these microbial changes during spaceflight affect aging in astronauts, though their exact mechanisms and extent remain unclear. Overall, the extreme conditions of space present immediate challenges to astronaut health with long-term implications for human well-being. The impact of spaceflight on the human immune system. Microbiota play important roles in modulating immune responses and influencing the aging process. Spaceflight-induced changes in microbiota composition potentially influence aging in astronauts. We studied how aging and spaceflight affect microbes in the body. Using genetic analysis, we tracked changes in microbe numbers and activity to find ways to protect astronauts' health and slow aging. Results could help design treatments to counter space-related health risks. Single-cell analysis revealed upregulated IL-6 pathway genes and elevated inflammatory markers, IL-6, MCP1, in astronauts, mirroring aging-associated immune changes. Shared immune-related gene signatures between spaceflight and aging suggest analogous immunological impacts, indicating spaceflight induces accelerated immune aging-like dysfunction through conserved inflammatory pathway activation. Using two-sample Mendelian randomization analysis, we found links between aging and specific microbes in the gut, skin, and oral cavity. Some microbes promoted aging, others slowed it. While GWAS data limitations suggest more research is needed, these findings highlight microbial targets for aging interventions. Aging triggers systemic microbial shifts across body sites. Gut analysis shows reduced beneficial and increased harmful bacteria, mirrored in oral/skin communities. Cross-site patterns indicate coordinated microbiota aging. These changes form host microbe feedback loops that may accelerate biological aging. Findings suggest microbiome-wide dysbiosis actively contributes to aging mechanisms, highlighting intervention potential. Analysis of 72 astronaut microbiomes revealed spaceflight-induced dysbiosis. Gut/oral/skin showed probiotic depletion and pathogen enrichment. Cross-body microbial covariation across taxonomic levels indicates systemic space environment impacts, highlighting microbiome-wide alterations affecting astronaut health. Spaceflight induces microbiota remodeling partially overlapping with aging. Harmful bacteria decrease post-flight, yet bidirectional shifts occur in some taxa. WGCNA and machine learning identified high-accuracy aging predictive modules, revealing cross-body microbial networks. Upregulated anti-aging-associated microbes suggest targeted microbiota modulation could maintain astronaut health through microbial host interplay. Spaceflight induces functional microbiota shifts beyond abundance changes. Metatranscriptomics revealed disrupted metabolic pathways linked to neurodegenerative diseases in oral/skin microbiomes, with altered activity of key enzymes. Integrated analysis identified covarying microbial functions affecting host physiology, emphasizing the need to evaluate both compositional and functional microbial alterations for comprehensive space health assessments. Overall, spaceflight alters the immune function of astronauts in a manner similar to natural aging. Aging alters the host microbiota across multiple body sites, leading to changes that may influence the aging process itself. Aging-associated microbiota in the oral cavity and skin exhibited significant changes after spaceflight. Spaceflight may potentially impact human health by altering microbiota function.